Everyone, welcome to another seminar of the International Tree Mortality Network. I am Bill Hammond from the University of Florida, and along with Bernhard, Adriani, and Nadine from the International Tree Mortality Network, we're really pleased to welcome you today to a seminar by Professor Brendan Choat from the Hawkesbury Institute of the Environment. He's going to give us a seminar, The Impact of Extreme Drought on Australian Forests and Woodlands. This is a topic that Brendan's been studying for a long time. He studies plant ecophysiology with a focus in particular on plant hydraulics and the impacts of drought on forest ecosystems. You can see some impressive examples of that here on the title slide of his presentation. He obtained his PhD from James Cook University in 2003 before doing a postdoc at Harvard from 2003 to 2005, and then another at UC Davis from 2005 to 2008. Brendan returned to Australia in 2008 to work as a research fellow at the Australian National University before moving to where he is today at the Hawkesbury Institute of the Environment in 2011. He was awarded an Alexander von Humboldt Fellowship for Experienced Researchers in 2010 and an ARC Future Fellowship in 2013. On a more personal note, I met Brendan long ago at a Gordon Research Conference on plant hydraulics, where then I was studying this phenomenon of tree death. His body of work has been inspirational and taught us all so much, and I'm very excited for you all to have the opportunity to hear this seminar today. So without further ado, Brendan, thank you for waking up. It's 7 a.m. in Australia right now. I'll let you take it away.
All right. Thanks so much, Bill. And thanks very much for inviting me, particularly to Bill and to Bernhard. It's great to be here. It's a real pleasure to address the audience. And this is, of course, a topic which is very near and dear to my heart. So I'll just start off, I think, by giving a little bit of a primer into plant hydraulics and water relations. I know that most people here will have heard this before, but in case there are people in the audience that have not heard it, I'll just go through it quickly. See. Okay, so plants don't have a contractile pump like the heart to move water up to the canopy. Instead, they rely on this unique but unstable transport mechanism in which liquid water is held under tension. This is really counterintuitive, but what it means is that this water can sustain a tensile force like a rope or a piece of metal. And this sounds very strange, but stretched water exists in many places in nature, particularly in drying soils. The advantage for plants of this mechanism is that they are able to transport very large amounts of water without the input of much metabolic energy. The downside of this is that liquid water under tension is subject to cavitation or vacuum boiling. And so cavitation is a phase change from liquid water to water vapor. You can see this little diagram here on the right-hand side of the screen. And when you have an embolism in a xylem vessel, gas will expand through that vessel until it reaches these very finely porous pit membrane walls here, or actually primary cell walls of the xylem. And it will be held here until the pressure difference across this barrier becomes great enough for gas to move through. And this process is known as air seeding, and it implies that the larger the pores in these pit membranes are, the more vulnerable the plant species will be to the spread of embolism. Okay, so this is perhaps better explained visually. And once again, on the right here, this is a cross-section of a eucalyptus stem, and this was generated using X-ray microtomography. So what you can see is the cross-section with the xylem here. You have the bark and the cambium, the phloem on the outside here. You can see embolized vessels in black here. And the functional vessels, the water-filled vessels, you can just see in this light gray color here. And so what I'll show you is a time series over which this stem was dried out. And you can see during that dehydration, the stem shrinks as you lose water content in the stem, but also that the number of embolized vessels increases very sharply as the plant dries down until you have virtually all of the vessels in that cross section embolized. And so this is obviously very bad news for the plant, because when vessels become embolized, they're unable to transport water. And when you reach a certain threshold, plants are not able to transport water to the canopy or anything else on the end of the branch. And we can show this graphically here. This is a diagram from a review that we published a number of years ago, where you have increasing water stress as the plant dries down. The stomata close, but even after stomata close, the plant will continue to lose water through the cuticle. So we have a cuticular conductance. The rate of water loss is much, much lower, but over sustained drought, and will eventually hit this point where you get a very sharp increase in embolism and the amount of embolized vessels, and that will lead to this progressive loss of hydraulic conductivity. So this is known as a vulnerability curve. It's typically referenced by a couple of points along the curve, psi 50, which is the water potential at which you reach a 50% loss of hydraulic conductivity, and this is kind of the industry standard for comparing species. There's, of course, a huge variation in this trait in terms of how much, or the water potential that the xylem can withstand before you start getting a sharp uptick in cavitation and embolism spread, and psi 88. And so psi 88 is a trait or a point which has been quantitatively linked to a minimum recoverable water potential, or a point of no return. So once plants go past this point, and there's no magic line, this psi 88 is very much an estimate, and it's certainly moved around a bit depending on the species and the dry down protocol that people have used. But somewhere in this zone here, when you reach the inflection point, it becomes very difficult for plants to recover from. So you get not only the loss of most of the conductive capacity of the network, but also you start to get death of the meristematic tissue. And at this point, the plant reaches a zone of kind of systemic failure, and is very, very difficult to recover. Okay, so the ultimate consequence of this, along with many other factors, are these mass dieback events. And so we've seen this increasingly globally as global temperatures and extreme events occur with more frequency, that you have these massive forest dieback events. So these are a couple of examples of this. This is a drought from California, which occurred now quite a while ago. This drought ended 2015, 2016, but this was a particularly bad one where over 100 million trees died. And you had many of the conifers in the Sierra Nevada mountains unable to recover from that drought. We've also seen quite significant droughts in Europe, where we've had very large areas of forest dieback. But even in areas that people consider very mesic and ever wet, such as the Amazon, we're also starting to see forest dieback and changes in vegetation structure, which have been linked to these severe droughts. And so one of the issues with climate change and increasing temperatures is that when you have a background increase in temperature, you'll have also an increase in evaporative demand. So when you have the increase in vapor pressure deficit, it means that droughts will set in earlier. They'll reach a great intensity, and they'll tend to last for longer. And this is really a way in which climate change is very directly and already impacting on a lot of the forest and vegetation systems around the world. And this is a very nice summary of this from a paper that Bill and colleagues published a couple of years back, showing all of the sites in which we have drought-associated mortality events occurring around the world. And you can see that they're really not restricted to the dry areas. They're occurring all across forest biomes, different forest types. We see in a lot of these very wet forests in Southeast Asia, South America, we have a number of dieback events recorded. So in Australia, we've also seen this in some of the tropical forests. This is some work published a couple of years ago by Bauman and colleagues. And this brings up another point, I think, which is important to consider. As opposed to these very extreme drought events, we have this dramatic broad-scale mortality that's very striking, and you see popping up in the news. We also have these background increases in mortality. So what this study showed is that with a background increase in VPD across a number of years, so this is a monitoring network in North Queensland, northern tropical Australia, in which forests have been censused, forests across an environmental gradient have been censused since the 1970s. And what they showed is this increase in mortality rate. It was coincident with the increase in VPD over that time period. And I think that's important. It's something that's much more difficult to tease out, particularly when you have a lack of good forest inventory data, as we do have in many parts of the world. But I think it's equally important to these very dramatic mass die-off events that we see from time to time. Okay, so now I'm going to step back and talk a little bit about the theory and some of the data that we've collected in the past. One of the things about measuring hydraulic traits is it's difficult and time consuming. It's one of these sets of physiological traits which you need to spend a lot of time in the lab. And so historically, we didn't have a huge amount of data to look at trends across biomes and across the globe. So back in 2009, along with some colleagues, we set about synthesizing the data set of vulnerability to embolism, water potential, along with a number of other hydraulic traits or xylem functional traits. And so this was the initial xylem functional traits data set, which Bill has led an effort to reconstitute and make freely available. So thanks very much for that, Bill, for the effort and others that has brought that data set forward again. So when we initially analyzed this data set, what we found is that this plot, we have Psi50. So this is the xylem water potential at which species will suffer 50% loss of hydraulic conductance. And the Psi-min, this is the minimum water potential that's been measured for that species. And we saw, of course, a very large range of vulnerability to cavitation, vulnerability to embolism across species and across environments. And you do see a relationship between, if you look at one of the climatic variables, something like mean annual rainfall and vulnerability to embolism. But the relationship is quite messy. It's not very tight, and you tend to get a lot of variation, particularly in the drier areas. This relationship between the minimum water potential and P50 is a lot tighter. And that's because plants have all kinds of adaptations that they can use to escape water stress during drought. Some have deep roots, some are drought deciduous, and it means that even if plants occur in areas that are very dry on average, they might not necessarily feel that in the same way as some of the plants that they're coexisting with if they have these water stress escape adaptations or mechanisms. But when you look at the water potential that they experience in the field, we see this very clear relationship and this very tight coordination between Psi50 and Psi-min. And this shows that the plants are very tuned to the water stress that they experience in the environment in terms of these hydraulic traits. And it really emphasizes what an important mechanistic trait the tolerance of hydraulic failure is. So the other thing that came out of this data set was this convergence of safety margins across forest biomes. So we have here looking at the safety margin, which is really the difference between your Psi50 and your Psi-min. So you can see there's a difference between the one-to-one line here, and particularly for angiosperm species in this data set, we saw a convergence across many different forest biomes and many different moisture environments to a very small safety margin. And we can see the same with a bit more space for Psi88 here. And what this means is that the plants are very tightly tuned to the environment that they're growing in order to, we think, in order to maximize carbon gain. So this is a trade-off in terms of the amount of carbon that they're able, or the amount of CO2 that they're able to assimilate, and the risk that they place themselves under in terms of hydraulic failure. And the big take-home message from this is that plants don't have a huge amount of physiological space in the climates in which they grow, and therefore vulnerable to a rapid change in that moisture environment. So when I came back to Australia, one of the things I noticed is that we didn't have a large data set of hydraulic traits. And so this is one of my first priorities, was to assemble a data set of hydraulic traits from species growing across different environments. And we utilized the Tern supersite network. So this is a network of ecological sites. In this case, the supersite network is dedicated to observation of ecosystem processes. All of these sites have eddy flux covariance towers, core hectare plots, which are censused every few years, along with a number of other environmental traits that are collected. And so it made, because it occurred across such a broad rainfall gradient, it made a perfect system in which to observe variation in hydraulic traits. And so this is the work of Jen Peters, who was a PhD student at the time, is now a postdoc. It's really fantastic work. Once again, showing this convergence across the network. In this case, at the wetter end, so these are species that occur both in wet tropical rainforest up here in North Queensland, but also in cool temperate rainforest in Tasmania. When we look at the minimum water potential that they experience, it's around the same. And we see this is actually a slightly smaller safety margin here for those species. Okay, one of the other things that we're interested in is the intraspecific variation in hydraulic traits. And this is some work that was undertaken by Rosana Lopez, who was visiting as a Marie Curie Fellow for a couple of years. And she looked at the variation within species in two species of Hakea. So Hakea is in the Proteaceae. It's a shrub. Hakea leucoptera grows across quite a broad environmental gradient in the arid zone in inland New South Wales. Hakea dactyloides grows in the moister coastal forests. And what she found is that even across this very broad environmental gradient, there was very little variation within species. So there was no systematic variation with rainfall across this environment. Instead, it was fairly constrained as a trait across the population, across those different genotypes or ecotypes. And what we do see is a separation between the two species here. And this is fairly similar to results that people have seen in the Northern Hemisphere with pine species, Pinus sylvestris and Pinus pinasta. In Bernhard's study, there was a strong relationship between P50 and the rainfall environment in which different populations of beech trees grew, but it was a fairly modest difference. And so what this means is that it's quite surprising in a way that there's not more acclimation of this trait. It might come down to the fact that the xylem is laid down, it's undergoes programmed cell death at maturity, and so it's much more difficult for plants to adjust these traits in the short term. But what it means is that if we are using incorporating these traits into models, we need to worry less about how this trait is acclimated between different moisture environments. That's not to say that we don't need to worry about it at all, but it does signal, the data that we have so far signals that there's not a huge amount of intraspecific variation in this trait, even when you have populations growing across broader environments. Instead, it seems like plants tend to adjust allometric traits like the Huber value or leaf area sapwood area ratio, or the root-shoot ratio in order to maintain their minimum water potential within the range that's set by their hydraulic traits, the vulnerability thresholds. And so this, of course, comes at a cost. If you have less leaf area deployed, that means that there's less area that's available for CO2 assimilation. And so it's not like there's not a trade-off there, but it's very interesting that as opposed to vulnerability to embolism being adjusted across these environmental gradients, but relatively stable as a trait, while you have these structural traits being adjusted in order to compensate for that difference across environments. So one of the other things that we've been working on with that in mind is the incorporation of hydraulic traits into models aimed at predicting the response of plants to drought and to water stress and to increased heat. So I've just got a couple of examples that we've been working with here. First, we have Hervé Cochard's model Sureau, which is a very complicated or very detailed whole plant model in which we have over 200 parameters represented, all of the resistances moving from the soil into the roots, through the trunk and up into the canopy and out into the atmosphere. And we compare this to the global dynamic vegetation models or these dynamic vegetation models, land surface models, which operate at a much broader scale. But because of the scale at which they operate, you have necessarily you can't include the amount of detail that you can in the whole plant models. So in most cases in these models, you have species aggregated into plant functional types. Grid cells are something like a kilometer square. And so we have to find some way to, if we want to include hydraulic traits, we have to find the best way to step between these very detailed models and the global or regional land surface models. And so this is something that we've been working on with an eye to how we can do this most efficiently. And I know that many of our other colleagues have. And it's really an important step, though, because the vast majority of these models do not have mechanisms to specifically deal with water stress-induced mortality. And so if we're going to be able to predict what occurs in response to these increasingly severe droughts, we really need to incorporate those kind of mechanisms into process-based models. Okay, and this is an example of the challenge. This is from a paper we published a couple of years back. And what we can see here is trace in water potential during the course of the drought of two species. And so the water potential the plants reach, and when they reach that point of the cavitation threshold, their point of no return, this is going to be determined by a whole lot of environmental traits, which are represented in these models: rainfall, temperature, humidity, soil type, and these structural and physiological traits: leaf area, rooting depth, stomatal response, cuticular leakage, water storage within the plant, the density of trees. And so this is the really challenging part. In a way, measuring our physiological traits is fairly trivial. It is time consuming, but it's something that we can do. The challenge of measuring when they hit that set point is much greater. And so this is what these efforts are directed towards, is really being able to take into account the most important traits that we have from the biology of the trees and environment and accurately predict the point at which it will reach that the point of no return. Okay, so the other issue that we faced is that a lot of our physiological measurements have been conducted in small potted plants. And so this is just a couple of examples of a small sapling in the greenhouse. This is some work at the Australian synchrotron. We got Jen loading a tortured plant onto the stage there. And so these experiments are absolutely essential to work out the fundamental physiology of the plant and determine the physiological mechanisms that underlie these relationships. But we run the danger of missing something when we're not working on mature plants that are growing in the field. And so up until recently, we we really lacked a lot of data to ground truth these relationships that we'd seen in potted plants. In Australia, from 2017 to 2019, we had a very severe drought. It was the worst drought on record across the continent in terms of the 2019 rainfall was the lowest on record. 2019 temperatures were the lowest on record. And if we look across the period from 2017 to 2019, we can see particularly in inland New South Wales and southeastern Queensland, down here in Victoria, we have the lowest rainfall on record and also the highest temperatures on record. And this led to this very dramatic and quite awful forest dieback, mostly in eucalyptus forests and woodlands that were distributed across this area. So these are evergreen trees. So all of the browning you can see here is leaf death. We have some conifers in here, which are still green. This is Calitris, a native Cupressaceae genus in Australia, which is more drought tolerant. We also had a very wide-scale bushfires that occurred after this. They were really precipitated by the environmental drying and the sustained high VPD. But these were forests that were not burnt. And it was at the time when this was occurring in the summer of 2019, 2020, we had bushfires raging everywhere. We had a lot of drought-induced mortality. It was difficult driving around to tell whether a forest had been burnt or whether it was just suffering from this really quite severe drought-induced dieback. So we set about studying this in the field. And so the first plot that we set up was in New England here. It's in one of the areas in which, one of the areas that was hardest hit by the drought. And we can see that we have some areas of trees that were still relatively healthy during the drought and many other areas where we had severe canopy dieback. And what we wanted to do was really to test this hydraulic failure hypothesis in the field on mature trees. And so we measured the native PLC. This is the PLC, which is, or the percentage loss of hydraulic conductivity, the amount of embolism which is present in the branches across areas of or populations that had relatively low stress, and that's kind of low levels of canopy dieback, high stress and dead and dying trees. And we measured the water potential of these populations. And so we can see for declining water potential, it was not possible to measure water potential on these trees, that we have declining water potential and increasing loss of conductivity. And in the dead and dying class, we have close to 100% loss of conductance in these branches. So this is really a nice confirmation of the hydraulic failure hypothesis, showing it occurring in mature trees in under an extreme natural drought. So we then wanted to look a bit more broadly. And one of the things that we've done is set up a citizen science project. So Australia does not have a coherent long-term monitoring network of forest plots. And this is really an impediment when we started trying to understand the impact of the drought, is that we did not have a network of forest plots where we could establish baseline data of number of trees that were still alive and the canopy health classes. And so Australia has it's a very large area, it's a low population. And instead of trying to fund this, we had set up a citizen science network back in 2017 in order to gather reports of dead trees. And of course, what we saw in 2018 and 2019 was this very sharp uptick in the number of reports we were getting. Each of these blue dots represents a report that a citizen scientist has submitted to the site. And we had a lot of reports concentrated in this area of New South Wales, also southern Queensland and around Victoria. And a lot of the people who were making these reports had lived in the areas for a long time and were saying things like, we've never seen anything like this in terms of the amount of tree death. So you have people living there for 50 years saying that. It's really a signal that things are changing a bit. So after we'd accumulated these reports, we then set about establishing a small network of drought plots. And so this is along this area here in New South Wales, from New England tablelands going down in the west into the southern highlands here. And all of these areas were badly affected. We have a gallery of images here. This is by and large eucalypt forest and woodland. As we move out to the further site west, we start to have more acacias present. But in all of these forest types, we saw very widespread canopy dieback. And once again, when we looked at the native PLC present in those populations as a function of the canopy browning, and so in this case, it's a little bit counterintuitive. We have 1 is actually the highest level of browning, 5 is the lowest level. And so when we have high levels of canopy browning within the trees, we also tend to have much higher PLC values. And across the network, if we look at the proportion of basal area, so this is the percentage of basal area in which we have almost complete canopy dieback. So these are trees which have lost all of their leaves, or the entire canopy is browned off. And when we looked between the drought, so this was data that were collected at the peak of the drought and post-drought, a trip that we did 8 to 10 months after release from the drought. So we had quite significant rainfall during this period. We can see that firstly, we have quite a lot of variation across all of the plots that we've looked at in terms of the amount of canopy dieback that's occurred, but also that there's a very tight relationship. And what this really says is that there was not much recovery across that 8 to 10 month period. And so there's a signal that it was not only canopy dieback, but also whole plant dieback. You do have to be very careful with eucalypts and a lot of the Australian flora. They have a strong resprouting ability. This is an adaptation to recover from fire and drought. And so we have it in the green points here, we have epicormic resprouting. So they have epicormic buds, which are buried deep in the bark, which can resprout. Really, you can have a tree which looks dead for 18 months or 2 years, and then it will suddenly spring back to life. We have non-epicormic resprouting, and these are species that tend to resprout from a lignotuber, so a big storage organ, which is located at the ground level. And you can, in this situation, you can have death of the entire above ground biomass, so the whole tree dies, but then it will resprout from the roots, resprout a new stem or group of stems from the roots there. There's not, it turns out, hard and fast rules for this epicormic versus non-epicormic resprouting. But we can see that across the groups, as we have them defined here, that there was a consistent high levels of mortality, which was not seen to recover in that 8 to 10th month period. We also looked at the relationship between 18 of these tree species that were included in the network between dieback and DBH, so the diameter at breast height. So we had in 5 of those species, we had a fairly clear relationship between DBH and dieback, indicating that smaller plants or smaller size classes within a species were more vulnerable to dieback than larger individuals. And this suggested to us that the smaller individuals had a less developed root system. Eucalypts, when they mature, tend to be quite deep-rooted and rely on water that is stored in deeper soil layers or on groundwater. And so in smaller size classes, it's easy to imagine that they had not developed that deep taproot at the point in which the drought hit, and so they were more vulnerable. But as I say, we only found this clear relationship in 5 of those 18 species, and we have had other studies, particularly examining the interactive effects of drought and fire, in which we've seen a kind of U-shaped distribution of mortality in response to those disturbance events. So we have both smaller individuals and the largest individuals, particularly those that have been previously damaged and have a fire scar. This is work from another colleague, Eli Bendel, that was published recently. So it's quite a complicated story in terms of life history and the size classes which are most vulnerable there. Okay, and then so we had a very awful drought followed by some of the worst bushfires that we've experienced in 2019, 2020. Since that period, we've had years of either very much above average rainfall or average rainfall. And so this is the rainfall deciles from September 2020 to August 2024. And we can see here that it's really flipped. And this is a characteristic of the Australian environment, that you have this very large variation in rainfall between years. So you have periodic extreme droughts followed by floods. And so we've very much moved in. We had 3 La Nina years in a row. We had very severe flooding in many areas. But it has also presented an opportunity for the plants to recover. And when we look at global temperatures, this is the land surface temperature anomaly. This is just for 1 month, but it's a good example of what you'd often see when you looked at these maps, with the northern hemisphere really suffering under very high temperatures, while Australia was sitting in this little pocket of lower than average temperatures down here. And so during this period, this has allowed many of the forests, but also some of the individuals to recover. And this is the epicormic resprouting that I was talking about earlier. So we have, in this case, plants that have these epicormic buds, which are buried in the bark. They've been exposed to severe drought and experienced a lot of dieback in the canopy, and then are recovering through epicormic resprouting here. And so when we looked, we revisited. So this is the 2020 trip that occurred 8 to 10 months after the drought had broken. And we can see that there's a lot of recovery. Certainly the ground layer and the grass has recovered. But if we look up in the hills here, we can still see that there, as particularly in the ridges, we can see that there are a lot of dead trees in there. And if you actually go under the canopy, so we can see here, this is an image taken from that hillside that I was just showing from a distance, and you can see that you have a lot of standing dead trees that died during the drought in some areas. In other areas, more significant recovery, and the canopy is looking a lot healthier. So the other thing that we undertook during this study was utilizing remote sensing. So we certainly need ground-based observations. There's no substitute for having ground-based observations. But if we ever want to understand what is happening at scale, we need to utilize the remote sensing tools, which have increasingly high resolution and more parameters included in them. So what we can see here, this is the normalized burn ratio, which fire scientists use a lot, but is a good indicator of canopy health and canopy moisture. We can see that at the point that we undertook our initial survey here at the peak of the drought, we have some of the lowest readings, and then there's significant recovery. And we can see these from the image chips. This is just an example from a couple of the plots we're looking at. So we go from February of 2019 through to either November 2019 or Feb 2020 there. You see this huge amount of browning across the environment, and then very significant recovery in the spectral indices that's occurred the year after that. And so I think this is one point that we have to be aware of, that when you see this recovery in greenness here, this can often be a little bit misleading in terms of what's going on with the trees. And this is something that's been consistently found across a few studies, although it hasn't stopped some people interpreting this recovery as recovery of the whole system. And so what we get in these cases a lot of the time in the short term, say 12 months after the drought or the fires, if you're looking at fires, is that there's this very rapid recovery of the understory. So we can see we have this rapid recovery in the grasses, probably shrubs. And if you're from the satellite's view and looking at these greenness indices, we see the environment greening up very quickly after that, because it's actually looking through the dead trees and the canopy and down into the understory layer. And so this can give a kind of false impression. Whereas you still have a lot of dead trees there. So in order to examine this in a bit more detail, we've actually visited these sites again a third time. This is work that's being undertaken by Ellie Nichols, who's a PhD student in the lab. And so this is what we can see. The average mean dead basal stem area. And so what I showed you before was that this was very stable from 2019 to 2020. We actually have seen some modest recovery in that in 2023, and this is after 3 years of above average rainfall or average rainfall. And so this is quite interesting now because it means that you either have plants that are coming back up or they're coming up into the size class that we're starting to measure, which is greater than 5 centimeters dBH, or you have some plants that have essentially come back to life. So we do see recovery there, but it's very staggered, and it's quite different to what the story that we see when we look at remote sensing. I should also just point out that while we've had a lot of recovery, we have not had a lot of incidents of severe drought on the East Coast. We have had a very severe dieback event in the southwest of Australia. So this is on the west coast. Perth is out here in the forests of this area. They've been exposed to significant drought in 2023 and 2024, and they've started seeing broad-scale dieback there. We have also had, and this is quite interesting in terms of what it means for the impacts of compounding stress. We've had short drought periods within that 3 to 4 years since the drought broke. So this is just showing rainfall deciles for a short period in June, September. It's really June, October during these years. And we can see once again that we had very low rainfall for this part of Australia and high temperatures. So during this period, this was last year, and by October and November, we were once again starting to get a lot of images of forest dieback. This was surprising to us because firstly, we'd had a couple of years, 2 to 3 years of above average rainfall. The environment was not particularly dry before it hit this point. And then we have a short drought which occurred in the winter in the spring. So this is an early season drought. The temperatures themselves were not particularly extreme, but in this area, this is in the photo that I'm showing you is from the north coast of New South Wales, so north of Sydney, up in this area. We had this very sudden drought-induced dieback occur, and so this is aerial imagery, or sorry, satellite imagery from PlanetScope, and we can see that in these areas of forest here, we went from a very green canopy to very, very significant dieback in a short period of time. And this is something, the degree of the rapidity of this dieback was surprising to us because this didn't occur as quickly during the last drought. And so what it's made us start to think about is the impact of compounding stress. So if you have environments that are still in the process of recovery, of undergoing this kind of staggered recovery from an extreme disturbance event, whether that be drought or fire or something else, and then the environment is hit by another stress event, even if it's not a particularly severe one in the context of what it experienced before, maybe it is more vulnerable to dieback. And I think with the shortening return interval of these disturbance events, as you have more frequent extreme events, you will get environments exposed to these disturbance events more frequently, and this will maybe make them more vulnerable and accelerate that pattern of dieback. And so, we're able to visualize this quite easily in terms of the extent of the dieback where it occurs. The spatial pattern in this case is quite interesting because we can see a lot of it is restricted to the ridges and the north-facing slopes. So these environments which are most exposed to higher loads of solar radiation and drying, and areas which have shallower soil compared to in the gullies where we have deeper soil. And we're just in the process now of looking at the spatial correlates where we had the most severe dieback. So if you look across these environments, if you survey where you have the most the highest impacts of drought, you can then use this information in random forest models, supervised classification to identify across wider areas where you have drought and what that is correlated with into what that's related to in terms of spatial variation. And in this case, what we're seeing is very much that north-facing slope, and the ridges are important areas where we've had significant severe bushfire in the past, and also areas that have been impacted by drought in the past. Okay, and so just I can see that I'm getting up on 46 minutes here, so I'll just finish up now. So in terms of looking at how plants recover, we're also interested in seeing the mechanisms of recovery. We have this comparison here between plants being able to refill embolized vessels versus the growth of new xylem. And this is once again some X-ray microtomographs showing cross sections of 2 eucalyptus stems. This one's a control stem, and this one has been exposed to a severe drought treatment and then been allowed to recover for 6 months. And what we saw in this case was that there was very little evidence or no evidence of embolism repair, and instead that the plants were able to recover by growth of new xylem. And so this is important because it's in what we've seen so far, it seems that embolism repair, which we once thought was a very widespread mechanism of recovery from severe drought, now appears to be less common. In fact, we've seen it in very few species other than those that make significant root pressure. And this means that recovery will occur more slowly and will be more expensive to the plant in terms of carbon resources that have been allocated. We can also see that in this new layer of xylem that has been developed during the recovery period, we have more frequent embolism. So this really, again, raises the question of how vulnerable these individuals would be to a second stress event or a third stress event. Okay, and I'll just quickly finish up here by talking a little bit about monitoring. As I said, we don't have much of a coherent monitoring network for forests in Australia. So one of the things that we are setting up at the moment is a monitoring network across the Blue Mountains, which is directly adjacent to where our campus is in Western Sydney. It's a World Heritage listed area, and it has allowed us to, when we have set up the transect, it's allowed us to cover a very large amount of environmental space going across these environments. So if we look at the mean annual rainfall and mean annual temperature, we have plots that are spread across those. We have now 30 plots, which are 30 by 30 meters with all of the trees over 10 centimeters DBH measured and tagged. And we're in the process of deploying instruments to continuously measure water potential, sap flow, and other physiological variables. And this is, I think, these kinds of monitoring networks, I think, are very important in terms of being able to evaluate how plants are responding to drought in the field, because they give you a level of detail in terms of the temporal resolution that we previously have lacked. I think particularly plant water potential has been something that's been difficult to measure remotely and continuously. And so we're now utilizing these microtensiometers produced by Floropulse, and they seem to be working in eucalypts. So we have this continuous output of water potential, and this is just testing at the face site. So anyway, I'm going to finish up now because I think I'm a bit over time. This is looking at, this is just a dashboard that we've developed in some of our demonstration plots showing the data that we've collected at the site. And we have sap flow and a range of environmental variables that are updated every 15 minutes. And we're hoping to roll this out to another 5 of those sites initially. Okay, and we will, with reference to what I was saying earlier, with the remote sensing, we really need to also get a better handle on the changes in vegetation structure that occur with drought. So instead of just looking at the spectral indices, if we're able to utilize LiDAR, which is increasingly accessible, then this will really revolutionize how we can look at the impacts of drought in terms of its impacts on vegetation structure. Okay, and I'll just finish up by acknowledging all of the fantastic people that I've worked with, former students, postdocs, and colleagues from around Australia and around the world, and all of the institutions and organizations that have helped to fund the research. And I will let that go. So if there are any questions now,
Yeah. Brendan,
yes.
first off, awesome seminar. Thank you so much. Wow. As someone who really enjoys learning about tree death, this was a great seminar for me, and I'm sure for our, I think at some point more than 50 attendees here in the Zoom. So we do have a couple of questions from attendees. I'm going to start with these and see where we're at on time. A couple of your hosts, of course, also have questions, but we will save those till the end because I guess you'll let me bother you later if I want, if I ask real nice. So our first question from the audience is from Vincent Rydell, who asks, you have shown tree mortality due to drought across the world. There were quite less points in the boreal area. Oh no, even if these areas cover quite a high amount of land surface area, how do you expect the situation there and how will it be in the future?
Well, it seems like the boreal zone, I mean, it is underrepresented, and I think part of that is geopolitics. But yeah, it's there's a couple of competing narratives which are emerging for the boreal zone. One is higher temperatures in the boreal zone will lead to faster growth of vegetation. That's possibly new species moving into that area. I think the other one is that if you have species which are, very much adapted to cold temperatures, you have an increase in temperature in VPD. You could get firstly a background increase in that mortality rate, but you could also get very significant drought-induced mortality events, which are compounded by forest fires. And I think we've already seen that happening in some parts of the Northern Hemisphere. So I think within those areas, spatially, it's going to be very complex. I think we'll have some areas in which we have increasing biomass in vegetation, probably new kind of faster growing species that are adapted to warmer temperatures moving in. And in other areas, we're going to have some very significant dieback events.
Thanks for sharing that. Oh no, and the question was mine because I put together the map that you showed, and yeah, I agree with a lot of what you said about geopolitics and data access. That whole map, I should just comment, was limited to published studies that I could access and authors who could verify locations of dead trees. Okay, our next audience question is from Deborah Coelho Silva, and it is, what would be the minimum time for a forest to recover after a dieback event? Is it more related to the area or the population itself?
Oh, I think there would be a huge amount of variation in terms of the species that were growing there, the intensity of the dieback event. I I would not want to hazard an answer, a generalized answer to that question. I think, what we've seen in Australia is that it's taken in the 3 or 4 years we've had, which have been, on the East Coast, relatively ideal, relatively ideal situation for recovery in which we've had either very, above average or average rainfall. We've seen the staggered recovery 3 or 4 years, and we're just starting to get back to the point now where, most of the forests are looking in good condition now, although, many of the patches that we surveyed, which were the worst impacted, have still not fully recovered and are carrying a lot of dead trees. So I would say, you're looking at 3 to 4 year periods if you have favorable conditions. And if you are, during that time, you have another large disturbance event, that will probably prolong that recovery. But in terms of a general statement, I think it's impossible to make that statement because you have so much variation in the not only the traits of the species that are growing in those forests, but also the density of the stands, the geography, all of these things which are important in terms of determining recovery.
Thank you for that. Our next question is from Yude Pan. Are those tree diebacks in Australia just a normal natural process, as tree species in Australia have been long adapted to dry conditions? Do you have some historical information for comparing today's dieback scale?
Yeah, so that's a really good point. What have we seen historically, and how does the current event compare? Periodically there are dieback events. Drought is very much part of the Australian environment. And so it is difficult to tease out whether these events are getting worse. Um, but what we saw in that event was certainly a greater amount of dieback than we had record of from previous droughts. We do have photos going back into the 1800s and the early 1900s showing dieback. And we do have a vegetation here which is well adapted to periodic drought and fire. But I think when you have these relatively rapid increase in temperature, relatively rapid increase in VPD, what we're worried about, even though you have species, vegetation classes which are superbly adapted to recovering from that, you really are starting to push the limits. And we have areas in which we've lost, particularly in things which are obligate reseeders instead of resprouters. If you have a short return interval on disturbance events like fires and droughts, and you start to get that vegetation knocked out and replaced by something else. So it's a good point, and I think no matter where you are in the world, you have to look at the context of forest dieback. But there's a lot of evidence accumulating now that the amount of stress that these systems are being exposed to is more than they have been in the recent past, and the levels of dieback that we're seeing are greater.
Thanks for that. A quick follow-up. This is from me. Is so then you were showing some plots that showed kind of like I think it was 2019 driest year on record. Would that include like the Federation drought? When I think of mega droughts that have caused like catastrophic multi-trophic collapse in large areas of Earth, that's one of the first that comes to mind.
Yeah, so that includes all of the climate records. That includes the Federation drought. And so it's averaged across the whole continent. So what you've got to remember is, whenever you have droughts occurring, most of the time they're fairly localized. So you'll have greater impacts in certain regions. And that's just saying that, across the entire continent of Australia, the in terms of that year, the rainfall that we had and the temperatures that we had were the lowest and highest on record, respectively. That's not saying that the ecological impacts were worse. It's very difficult now. There have been attempts to kind of quantify what happened during the Federation drought, just based on essentially kind of media reports and narrative during that era. But certainly in terms of just the meteorological data, that was the worst that we've had on record. And I think a lot of it comes down to just this increasing VPD. And when you have the severe end of the drought really start to set in.
Cool. Thanks for that elaborating on that, Brendan. The next question is from Daniel Balatsengoy. Says, you work in an environment with trees that really like to be thirsty, hot, and sometimes burnt. But really the ones you study are the offspring of the survivors. With this in mind, my question is, with an increased focus on real-time ecophysiological monitoring, what observations and tests are you really excited on seeing in the near and long-term future?
Okay. So I think that there are actually there's quite a lot of surviving mature individuals. So we're not just looking at the offspring. So I guess you're alluding here to work on seedlings and saplings and trees that are kind of in the earlier stages of development.
They may be, but also they might be indicating survivor bias. I don't know. Like if we go and study the ones that made it.
Yes, yes. So I mean, in terms of ecophysiological monitoring. The thing that I'm most interested in is getting more insight into the level of water stress that those individuals are exposed to. And I think, looking at the difference between things that are in an early life stage versus mature trees that have survived is really important. I mean, what are the implications for trees that have not established a deep root system yet, but are hit by, a severe drought? And I think we really lack, it sounds kind of boring. I just want to measure water potential, but, we, that minimum water potential that I showed from the plot at the start of the talk, that's just the water potential whenever someone could get out there with a pressure bomb and measure it. And it's really, just a snapshot. I think we would have so much more information, so much more data that would of, enormous value, particularly to the modelers, if we have continuous output of water potential and we can really see, when stomata start to close and when plants hit those points, how much they bounce backwards and forwards between zones of, being close to going past the point of return and then recovering. I think that, those data are super important and that we don't have good records of that at the moment.
Uh, we don't. I'm probably biased, but I'm also just really want to measure water potential. So agree with you. Brendan, that's the hour. So we have a few more questions. We're gonna go ahead and end the seminar there. I'll reach out to you with some of mine later. Because of the format, we can't see all of the attendees here, but I'm sure their applause might reach you all the way around later this morning in Australia. I've really enjoyed it. Thank you for your time and sharing your expertise with the International Tree Mortality Network and our whole audience.
Yeah, well, thanks very much for inviting me. It's an enormous pleasure to give a presentation here, and I'm really looking forward to. Well, thanks also for the people who've stayed up till 9 o'clock at night. I hope you're still enjoying your beer, and for those who are kind of on their lunch break or taking a bit of time out in the afternoon. Yeah, it's an enormous pleasure, and I'm hoping to get to that Xylem International Conference in Sevilla next year and to see some of you in person again finally.
That'd be awesome. Look forward to seeing you in Sevilla. Take care, Brendan. Take care, everyone.
Cheers.
Bye bye.
Goodbye.